Hey curious minds, welcome back to Cellcrafted. Today we are diving into the microscopic world of the earliest life forms on Earth, prokaryotic cells. Let's explore their structure and functions in detail. Let's begin. What are prokaryotic cells? Prokaryotic cells are the simplest and most ancient forms of life. They are the smallest types of cells found in nature. They lack a true nucleus and membrane-bound organelles. Their size typically ranges from 0.1 to 5 micrometers. These cells are usually unicellular and microscopic and includes two major groups, bacteria and archaea. Structure of prokaryotic cells. Let's break down the structure layer by layer. Cell wall. In prokaryotic cells, the cell wall is an essential structure that provides structural support, maintains shape and prevents lysis due to osmotic shock. The cell wall of bacteria is composed of peptidoglycan. Archaea have different cell wall structure. The cell wall acts as an anchor points for surface structures like flagella and pili. Beneath the cell wall is the plasma membrane, also called as the cell membrane, which is composed of phospholipid bilayer with embedded proteins. The plasma membrane is a semi-permeable barrier that controls the entry and exit of substances involved in respiration and transport processes. It is the major site of respiration and ATP synthesis since prokaryotes lack mitochondria. It is also involved in the synthesis of lipids, cell wall precursors and other macromolecules. Though structurally simple, it is functionally versatile, compensating for the lack of organelles. Cytoplasm. It is a gel-like, semi-fluid substance that fills the inside of a prokaryotic cell. It lies between the plasma membrane and the nucleoid region. It is composed of almost 80% water, dissolved salts, ion, sugars, enzymes and proteins. Prokaryotic ribosomes and genetic material resides in the cytoplasm of the cell. The cytoplasm maintains the internal structure of the cell. It is the site of various metabolic activities because it contains all the essential enzymes. Next region is the nucleoid. It is an irregularly shaped region in the cytoplasm of prokaryotes which contains the circular DNA, which is the genetic material. The nucleoid is not enclosed in a membrane, thus facilitating DNA-cytoplasm interactions. The primary function of nucleoid is the storage of genetic information. The nucleoid holds the primary genetic material, which is a single circular chromosome of DNA. This DNA contains all the information necessary for the cell's functions, growth and reproduction. The DNA in the nucleoid controls all the major cellular activities such as gene expression, DNA replication and protein synthesis. During binary fission, the nucleoid DNA replicates to ensure that the daughter cells inherit a copy of the genetic material. This process occurs directly within the nucleoid region since there is no nucleus to contain it. The DNA in the nucleoid region contains genes that are transcribed into RNA, which is then translated into proteins by ribosomes in the cytoplasm. The prokaryotic DNA is not associated with histone proteins, but some archaea do have histone-like proteins to help in DNA packaging. The next structure is the plasmid. Plasmids are small, circular, double-stranded autonomously replicating DNA molecules that exist separately from the chromosomal DNA in prokaryotic cells such as bacteria. Plasmids are much smaller than the chromosomal DNA, ranging from 1,000 to 20,000 base pairs. They are not essential for the cell's basic functions but carry genes that can provide additional advantages to the cell such as antibiotic resistance, enhanced virulence and metabolic capabilities. Plasmids can be transferred between bacteria, allowing for the rapid spread of traits like resistance. Ribosomes. Ribosomes are tiny, dense structures made up of ribosomal RNA and proteins. They are not membrane-bound and are found freely floating in the cytoplasm of prokaryotic cells. They are known as the protein factories of the cell because their main job is to synthesize proteins. Prokaryotic ribosomes are 70S in size and is made up of two subunits, a small subunit, 30S, and a larger subunit, 50S. Prokaryotic ribosomes are smaller and lighter than eukaryotic ribosomes. Flagella. Flagella are long, whip-like appendages that extend from the surface of prokaryotic cells. Their primary function is mobility. A flagella has three main parts: a filament, hook and basal body. Pili. They are hair-like projections found on the surface of prokaryotic cells. Pili is made up of protein subunits called pilin. They help in conjugation. Some pili also assist in attachment to host surface or cells. And that brings us to the end of our deep dive into the structure of prokaryotic cells. Let's quickly wrap it up in a nutshell. Prokaryotic cells are the simplest form of life, but they pack everything they need to survive, grow and reproduce. They do not have a true nucleus or membrane-bound organelles, but they are still incredibly efficient. It has a cell wall that gives the cell its shape and protects it. Plasma membrane controls what goes in and out. Cytoplasm, where all the internal action happens. Nucleoid holds the cell's genetic material. Plasmids are small extra bits of DNA for bonus traits. Ribosomes build the proteins the cell needs. Flagella help the cell swim through its environment. Pili help the cells to stick to surfaces. If you find this video useful, don't forget to like and subscribe for more clear and simple videos. Thanks for watching. See you all in the next video.